Here's a defense system. Try to get in this house without the alarm going off. Okay, they throw just a little HIV stuff in here. Oh, sorry, I'm on the wrong slide. They, the word acquired is what the A in AIDS stands for. So that's what threw me off. So here we're now getting to the specific defense system, the one that used to be called the immune system specifically because it leads to immunity. But now this is again called the acquired immune response. It involves those white blood cells that are called lymphocytes. And again, there's two kinds of those. And they are going to get triggered into action by bad guys like bacteria and virus and fungus, the most common pathogens, by transplanted cells, and even your own cells that have turned cancerous. They can all be recognized as foreign, bad guys, or a common term here is non-self. And what this acquired immune system does is to attack non-self things. So these lymphocytes, again, weren't specifically named, except these, as we'll see in a minute, are specifically named in that they're the ones that produce the proteins called antibodies. They didn't mention these T lymphocytes at all, and I am going to just a little bit. But these are specialized white blood cells that circulate around. Again, a whole bunch of them are in your lymphatic system. Your spleen is part of your lymphatic system. And this is where a lot of bad guys bump into them. But they circulate around, again, through blood vessels and lymph vessels, but also just through your tissues, like through the tissues of your kidneys and stuff like that. These things are squirming all around. So here is a key conceptual thing, and this overlaps with lots of ideas about how important the shape of a molecule is, right? Every organism has unique molecules on the surface of its cells. I just read the objective to you, right? That means you have different molecules on the surface of your cells than the person next to you. Some unique different ones. Most of them by far the same kind. But you have unique ones. Every single human being, which we're focusing on, and every single dog has some unique molecules on its surface. So the molecules that stick out from the surface, most of them are sticking out from the surface of these cells, good guys, pathogens, bad guys, cancer cells, good guys that have turned into bad guys, are called antigens. We have three anti-words involved with being sick that you need to keep straight. Antibiotic, antigen, and antibody, a word that sounds a whole lot like antibiotic. Here's one thing to remember. Antibiotics are not made by you. They're made by some other organism or now in the lab maybe, and we use them as medicine. Antigens and antibodies are part of this story of your defense system that we're talking about. Okay, so these B and T cells, when they grow up, they shuffle some genes around. This is transposon stuff. They shuffle some genes around, and every single different one comes up with a different combination of gene parts that makes a protein that they then put on their surface and call a receptor. So I'll just do a simple thing here. If this is one of them and its receptor is shaped like that, this might be another B or T cell and it has a receptor shaped, I'll make it like that, right? These are proteins. They're not nearly as big as I've drawn them in reality in relation to the cell size, right? But these are the receptors that some kind of antigen might fit into if it's the right shape. So I'll make a bad guy here. We'll call that a bacteria. Bad guy, pathogen, okay? If it has molecules on its surface that are shaped like this, right, you can see that those two puzzle pieces fit, and this is how so much of biology works because puzzle pieces fit. When this does connect here, this is by random motion, right? They bump into each other, right? Then that activates this B cell. What does that mean? If you think about it for a second, you can get it. That means this connection triggers a signal transduction pathway to make this cell use different genes to make different proteins to make different things happen. But very specifically, that same bad guy with the kind of triangle antigen, right? That would be the antigen part, right? This again we call the receptor. That particular bad guy, pathogen, bad guy, is not going to trigger this specific cell. It's got to bump into the right one. B cells secrete proteins called antibodies. Here they, oh, my fault. They did mention T lymphocytes, but this I think is the only place they mention T lymphocytes, right? After, and again, the standard level syllabus promises to not ask you about certain types of white blood cells. The higher level syllabus does. So what do B cells do? B lymphocytes, when they're activated, they multiply. How? Again, by mitosis. What's that make? Identical cells, clones. They make clones of two different varieties. Plasma cells, right here it says, are active. They're actively making by protein synthesis and then secreting these proteins called antibodies. Memory cells just hang around. But back a few, and it's coming up again, you're going to see that the key to immunity lies in the presence of these memory cells. What do antibodies do? We're going to, I think, come to this on another slide too with a little graphic. They aid in the destruction of bad guys, pathogens, directly, kind of, as we'll see. So here is kind of what I did. The B cells all have different molecules sticking out of their surface. You can't see it too well here, but what's different about them is just the tip of this Y part. Now, for B cells and only B cells, these receptors are also the molecules that we call antibodies. Look, see how they're Y-shaped? And they are Y-shaped proteins. So we call them receptors if they're doing the job of recepting. You can see here they're doing the job of connecting to an antigen molecule. But when they make them and then release them, then we call them circulating antibodies, So here we go. This process is called clonal selection. Out of all these different B cells, the presence of a particular bad guy's antigens has triggered one of them to be selected. That's how the selection works, to multiply yourself and make a whole lot more of you. I really don't need that either. So, sorry. Antibodies aid in the destruction of pathogens by attaching to them. So they have this labeled antigen. Really, this is more like the antigen part. Let's label this the pathogen, the bad guy, like a bacteria cell or a virus. When antibodies stick to them, why do they stick? Their specific shape. Notice that this bad guy has several different antigen shapes that increases our chances of being able to fight against it. And when it's clumped up like this, it makes it an easier target for white blood cells, because these clump with each other, these antibodies stuck to antigen cells, and at the end of the day, what's going to kill this bad guy is being eaten by white blood cells. So we go back to that. Antibodies don't kill them. Antibodies just slow them down, in a sense. The T cells that are called cytotoxic, they don't kill them. They kill your own cells. The B cells don't kill them. It's phagocytes that end up killing the bad guys at the end of the day. So there's two different kinds of T cells, but they don't even go into what those two different kinds of T cells do. But I'm going to do just a little bit, especially when we get to HIV. Here is the AP syllabus as it still exists, talking about cell communication. and saying that we might choose this example to describe to you the direct contact of a cell that's holding an antigen up, an antigen presenting cell, and these kind of T cells, or maybe they could have put B cells in here too. Like this, okay? So here's what happens for those kind of T cells that we call cytotoxic. They're like those natural killer cells we saw before, but they're very specific killers. But like the natural killer cells, what they kill are your own cells. This white blood cell, once connected to one of your own cells that has been virus infected, the virus is in here, and all these are parts of the virus. Therefore, they can act as antigens that lets our immune system recognize the virus. Our cells have evolved a way to grab these, hold them out, as if to say, hey, anybody recognize this shape? Because I'm in trouble, I could use some help. If a cytotoxic T cell with the right shaped receptor bumps into it, it triggers this, again, signal transduction pathway, to make a molecule that pops the membrane of this cell. All this stuff flows out, and this cell now is no longer making viruses, and so it helps in that way. Doesn't kill the virus, doesn't kill bacteria cells. In fact, these cytotoxic Ts don't have any effect on bacteria cell infections, but they can also kill cancer cells. So if you'll pretend that this, instead of being an infected cell, is a normal body cell that has mutated to become cancerous, cancer cells also throw out different kinds of molecules than normal cells, and cytotoxic T cells can take care of them as well. Here's where the macrophage, besides non-specifically gobbling up bad guys, part of the innate immune system, they can also grab pieces of that bad guy that they're digesting for lunch, hold them out, present them, and the kind of T cell called a helper T cell can connect there, and the helper T cell then starts to send out chemical messages. It can be, by analogy, related to the general in a warfare battle, which this is commonly framed as a warfare kind of thing. The general just gives orders. The general doesn't do anything like this one does. It doesn't make antibodies, which would be like weapons like the B cells do. The generals give orders to these kind of cells and the B cells to, hey, step up your game, do mitosis, make more of yourself. Let's get a handle on this specific bad guy with this specific antigen and antigens. So this is not in the syllabus either, but they gave names, and I don't know why they chose this name. I'm sure there's a good reason. The humoral response is the one that's all about making antibodies. The cell-mediated one is the one about cytotoxic T cells killing your infected cells. I really don't like this term because all of these responses and processes are mediated by cells. So anyway, so this graph is really good. And here we have something that I think we already saw, but if not, here we're going to see it again, or for the first time, whatever. But we did already say that activated B cells, first of all, are called plasma cells. Activated B cells, so I'm going to go back. In this description, activated B cells means they've bumped into the bad guy, and what they've been activated to do is mitosis. So they divide, and they divide into a kind of B cell that we call a plasma cell, and it's the active one. That's why I tripped up on that, right? It is the active one that is actively making antibodies, right? But some of those specific B cells with their specific receptor are just going to hang around. But now you have a whole lot more of them. And so when this bad guy bumps into you the second time or the third time or whatever, you have a whole lot of specific fighters against it. And this is the heart and soul of immunity, that it's memory cells that give immunity more so than antibodies. And we're going to see that in a second. Okay. So the helper T's are very key again, in that when the helper T's get activated, they send out these chemicals called interleukins. They're a specific type of cytokine. That word we said already. They are signal molecules. These cells, this helper T cell, T helper, TH, is now talking to these cells, cell-to-cell communication, at a distance, not by direct contact. These cells are talking to each other by direct contact. Okay, so you have that the kind of cell, that kind of AP, syllabus cell communication stuff, which really is none in the IB syllabus. Really should be. So anyway, these activated B cells making antibodies, that's called the humoral response, and these activated T cells killing infected cells is what's called the cell-mediated one. But here they do want you to know, and you saw this already, that B lymphocytes can be activated by T lymphocytes, specifically the helper Ts, but they can also activate these other kind of Ts. They just left that out of the syllabus objective. Here we go. Okay, so I probably shouldn't even have left this one in. So here's the cytotoxic Ts again. I've already showed you that. They are latching on to a specific, an infected cell with a specific virus, and they are releasing this chemical called perforin, which is going to perforate, which means pokes a hole in that infected cell. Already showed you that. Can also affect, like we already said, against cancerous cells.